Boston of all the experts who did a lot of sequencing, and they concluded that the genome does not have most of the answers that we are looking for. Then where is it? And that's what I'm going to try and introduce to you. They went back to the drawing board and they looked at some things they've observed for long periods of time. For example, differences in variations, and a more recent terminology that explains most of these intangible variations is epigenetics. What is epigenetics? Epigenetics is a science that is picked up in the last 2 decades quite a bit, and it is something to do with the study of changes in gene expression that are not linked to DNA sequence changes, unlike what we believed in the first half of from 1950 to 2010. What we believed everything is linked to DNA sequence changes, but this science is a slightly different science. It's picking up. There are now hundreds of thousands of papers on epigenetics. But bacterial epigenetics is still in its infancy. We have perhaps only about a couple of dozens of papers on bacterial epigenetics. I'll try and introduce 1 or 2 slides on what is epigenetics. I think I'll just skip some slides. So if you look at this, we all know that our DNA is very long. It's compacted using histones. And these histones have tails, and these tails can have additions, which are called epigenetic tags. It could be a methyl group, it could be an acetyl group, it could be a phosphate group that can compact the genome or open up the genome, and that changes gene expression big time. So these tags are called epigenetic tags. They can be on the histone, they can also be on the DNA. How is eukaryotic epigenetics different from prokaryotic epigenetics or bacterial epigenetics? Bacteria do not have histones, so all the changes, all the tags that you are going to see or you're going to look out for, they have to be on the DNA or RNA of the bacteria because they do not have histones. Now, in terms of bacterial epigenetics, what do we know? Typically, you all know restriction modification systems. You have a methyltransferase, you have an enzyme that will cleave it, so this is a defense mechanism that most bacteria have. But apart from that, there are some methyltransferases that are not part of the restriction modification systems. They're called orphan methyltransferases. Now, these orphan methyltransferases are more and more being characterized these days, and they have a very important role in gene expression. And bacterial RNA can also have adenine methylation. It's called M6A, which is an adenine. All the adenines or some of the adenines have a methyl group tag to it, and that can change a lot of things for bacterial RNA. It can determine where it's localized, what is the half-life of the RNA, what kind of rate of protein synthesis it will have. It can impact a variety of things for the bacteria. Now, this science is still actually just about beginning in terms of bacteria. We actually reviewed all the literature, and I'm just going to show you a few things from that, and we have some data. I'm not going to show you the data, but I'll quickly introduce you to some basic concepts in the field. So if you look at, if you take a colony, grow it in a broth, and subject it to the MIC of antibiotic, there will be no growth, it will all die. If you subject it to small concentrations, sub-inhibitory concentrations of the antibiotic, slowly there is some sort of selection that happens. Some bacteria survive. Now what happens if you withdraw this antibiotic? If you withdraw this antibiotic, in 1 or 2 generations, they all become susceptible again. And so the whole school of thought is that this cannot be genetic. It cannot be possible that a genetic mutation happens in a generation, it's lost in the next generation. That is very unlikely. So there is now more and more evidence that's supporting that this is all epigenetic, and epigenetics can also be inherited during cell division. Now, this is a more likely explanation. You have bacteria which are susceptible, and when you have the antibiotic stress, it can have the tag, the epigenetic tag shown here in orange, and the tag can allow some changes in gene expression that makes the bacteria resistant, and when the antibiotic is withdrawn, the tag can simply go away because epigenetic changes are so dynamic. If you take our WBCs in the morning and evening, we have the circadian rhythm or the sleep-wake cycles. Our DNA methylation is so different in our WBCs in the morning and evening. That's how dynamic epigenetics is, and that can also allow changes in bacteria very quickly, unlike genetics, which is which is much more slower and mostly not reversible. Okay, so in terms of epigenetics, a couple of interesting studies. So I think many of us would know the DAM positive strain or the DAM negative strain. It is an adenine methyl transferase that's commonly present in most of the E. coli. So one laboratory took a wild type strain that has the adenine methyl transferase. We also took another strain which is identical except that it lacks the adenine methyl transferase that's shown on the right side. And they subjected both strains to antibiotic stress. Now here's what happened. On the left side of your cartoon, if you look at it, that's the wild type strain. On the left side, excuse me. So anytime you have these stress, you have error-prone polymerases which are made in large quantities inside bacteria, and they create a lot of mutations. Unless these mutations are fixed, the bacteria will die. So because there is adenine methyl transferase or the M6A methylation happens on the left side bacteria, that's the wild type bacteria, the mutation can be repaired by the repair machinery which only senses the adenine methylation and then it can repair it. So if that is absent, therefore polymerases will actually kill the strain, and that's what happens to the DAM negative strain which do not have the adenine methyl transferases. When you subject it to antibiotic stress, they get killed. So survival during antibiotic stress, epigenetic tags on bacteria, very important. This is a Nature Genetics paper. And similarly, there is a role that has been shown for cytosine methylation of bacteria. Typically, cytosine methylation in humans, it suppresses gene expression. The DCM methylation in bacteria, in E. coli, that is also known to suppress the expression of some transcription factors. It's also known to suppress or regulate the levels of efflux pumps within E. coli, and DCM levels, the presence of cytosine methyltransferases can impact resistance versus susceptibility to bacteria. So the other very interesting thing about the interplay between genetics and epigenetics comes from the ability of methylated cytosines to undergo a very rapid rate of mutation. So methylated cytosines become thymidines by a process called spontaneous deamination, which happens in humans and bacteria, and they cannot be repaired easily, and they remain thymidines. So C to T mutations happen because of methylation in bacteria. This contributes to evolution of bacteria. This contributes to survival during antibiotic stress. Excuse me. Just the allergies from the Delhi air. Okay, so on the right side, what I have is how methylation of adenines is very important for DNA repair and survival during antibiotic stress. Now, how do we look at these epigenetic tags, these methylation of cytosine or methylation of adenine? How do you look at it in the laboratory at single base resolution? 3 million, 5 million bases of bacterial genome, and every base you know whether it's methylated or not, thanks to a couple of technologies that have come up in the last 5 to 10 years. One of them is called the PacBio technology. I think many of you may be familiar with it. And this works by the principle that whenever a base is methylated, the Taq polymerase takes much longer, almost 5 to 6 times longer, to add a complementary base opposite to that. And this kinetics is measured by this method. So the other technology is Oxford nanopore technology. Excuse me. So here you have a membrane with a lot of pores, and DNA is just ratcheted through the pore, and there is a current flowing through the membrane, and each base will disrupt the current differently, and that's what is measured. And methylated cytosines and unmethylated cytosines, they disrupt current differently, and that can be measured. So you can have single base resolution of the entire genome, whether or not there is methylation. You can get that information in less than a day using these technologies. Last couple of minutes, I'll just show some data from our laboratory, not in great detail. We tried to look at some Staph aureus and looked at some methylation of Staph aureus. We used some quality control methods, and then we found almost 9,000 adenine methylation sites in Staph aureus. Nobody has reported adenine methylation in Staph aureus. We also found differences between MRSA and MSSA in terms of methylation sites. And these methylation sites were particularly enriched in the non-coding regions or regulatory regions of Staph aureus. We also identified the consensus motive of the site that's shown inside that circle. We validated it by different methods. Very interestingly, we also mapped these sites. Where are these sites in the genome? They appear to peak right before the gene body, which is in the promoter region, or they appear to be at the 3' end of the gene body, which is just immediately downstream. Thank you. Downstream of the gene body. So these are the sites where the methylation happens in Staph aureus. This data we are in the process of submitting it. We also looked at hypervirulent Klebsiella. So I think a lot of you know classical and hypervirulent Klebsiella. It's a pathotype, and they have very big differences in terms of virulence factors, in terms of causing multiple organ infections, and more recently, hypervirulent Klebsiella has also become multi-drug resistant or extremely drug resistant. So we've sequenced almost 9 each of classical and hypervirulent Klebsiella, and we find almost 10-fold differences in adenine methylation levels in hypervirulent Klebsiella. We've also done more work in terms of triclosan adaptation and the role of methylation. We find that ability of E. coli to have a methyltransferase or if it does not have a methyltransferase, it matters a big deal in terms of its ability to acquire resistance when it gets acclimatized in terms of the presence of a biocide such as triclosan. I'm not showing all that data. I think that's about my time, and these are just my collaborators and funding over the last 10 to 12 years. And thank you all for the organizers for giving me an opportunity to be here and give this talk. Thank you.
Thank you, Dr. Perumal. Next invite